Okay, in the world of amino acids, there's a couple of ways that we can analyze for them. There's a couple of tests that we can do. As you would expect, protein chemistry is very important. Proteins are very complicated, and we'll see that when we get to that point. But, when we look at these amino acids and how they begin to take shape and how they begin to fold on top of themselves to make these massive proteins that do a specific job, well, protein chemistry is very important. We kind of want to know what those proteins do. We want to know how they do their job. We want to know what amino acids are actually involved in them to get them to do their job. So there's this huge push and this huge research and development end on protein chemistry. We really are starting to analyze these amino acids and trying to figure out, well, what is there, what's the order of them, and so forth. There's three major ways that we can separate amino acids from each other because that's part of the job, right? We know that they're there. Well, how can we separate them? How can we isolate them in order to maybe do some more tests with them? Well, there's three things that we can do to separate amino acids or to isolate the amino acids. And number one option is what we call electrophoresis. So electrophoresis. This basically means movement with electricity or some type of voltage difference. So electrophoresis, we will talk a little bit with amino acids, but it's also going to come back to the DNA portion of the class as well. That's later on in the semester. So electrophoresis we'll explain now, and we'll look at the method that's used to separate the amino acids. But keep in mind that electrophoresis does come back around. We do a slight modification of the process that we're going to describe here, though, and we treat it a little differently when it concerns DNA because it's a different type of molecule, right? So of course that we should probably treat it a little differently. Okay, so electrophoresis is option number one. Option number two is TLC. So for those of you in instrumentation, well, you probably have taken some TLC labs, and you've probably have analyzed something on a TLC plate. So TLC, of course, stands for thin layer chromatography. Again, that is thin layer chromatography. So TLC is another way that I could isolate these amino acids. I could take a mixture of amino acids, I could kind of mix them together, spot them on a TLC plate, and hopefully I would get some separation and I would see a couple of different dots or spots on my TLC plate in general. And number three, another way that I could separate these amino acids, because we keep talking about charges, and if I look at maybe the instrumentation side of some analytical chemistry, well, I would see ion chromatography as a suitable way to separate amino acids from each other. So this is not uncommon. I can take a series of organic acids, so I can take a sample of wine into the lab, and I can inject this wine on an ion chromatography instrument, and I could get some separation of organic acids. So this is very important maybe to take a look at the quality of the wine and how it's breaking down and how it's degrading, and all of that is playing off of the acids, which are charges because I'm looking at charged species depending on what kind of solution I'm putting it in. The same thing's happening here. It's just amino acids now because these things are charges, because they can have a full positive or a full negative on them. If you control the pH, you will be able to get it done. So ion chromatography is another suitable method to analyze and separate these amino acids from each other. So we're going to talk briefly about each one of these, and this video is going to focus on electrophoresis. I might not cover the entire thing, maybe if I'm lucky I can, but we'll take a look at least at electrophoresis first, and we'll see how far we get. So electrophoresis, we said, was basically separating. So we're separating things by charges, and we're using a voltage in order to get that done. So we first have to have an electrophoresis chamber, and this is basically a plastic slab. And this chamber is typically kind of like a box. So yeah, excuse art, my art 101 class. Again, I probably barely made it through. But this is a box. We call it an electrophoresis chamber. And inside of the box, what we do is we put a liquid solution. And that liquid solution is a buffer. So we fill this well, this electrophoresis chamber or this well, with this buffer that we have. With the electrophoresis, it can come in a couple of different forms. Some forms you will see one box, and in other forms you will see two boxes. So just for simplicity, let's just say that we have two boxes here, and you could probably understand it a little bit better if we do it that way. So here is one box, and then here is the second box, and I've put a buffer into each one. So I've got a liquid solution over here and a liquid solution over there. What we do is we put a charge distribution on it. So it's almost making like a galvanic cell. So think of that electrochemistry back in the day with General Chemistry 2. We went through this whole thing about galvanic cells and how to make batteries. The same kind of thing happens here. One end of the chamber is positive and one end of the chamber is negative. So I'm just going to put maybe the negative end over here and the positive end over here. Okay, so when we do that, we get this charge distribution. We get half of the well negative, half of the well positive, and we have to begin separating the amino acids somehow. And what will go on is that we typically will have a sheet of paper or something involved that we can kind of spread across both of these chambers and dip it into the actual buffer solution. And then what goes on is that we, on this sheet of paper, have spotted our mixture. And I'm just going to put my spot here. Okay, so there is the spot that has my component on it, and that component has multiple amino acids. And what begins to happen is that we start to see a voltage distribution on both ends. We start to see some movement. Some of the amino acids are positive, and they will migrate to the negative end. And some of the amino acids are negative, and we migrate them to the positive end. So just like ends of a magnet, well, opposites attract, right? Okay, so how do we know what happens here? Well, let's say that I've got 3 amino acids, and the amino acids that I have, let's say, is arginine. Forgot to take it off the white. So arginine, and this has a pi value of 10.76. And then let's say that we have alanine, and alanine has a pi value of 6.02. And then let's say we have aspartate, and aspartate has a pi value of 2.98. Okay, so here are 3 amino acids that we've pulled from the table. And I've calculated the PIs for each one of them. This is why we learned how to calculate PIs, because the PI values will tell you what they do. The PI values will tell you how they begin to migrate. So the PI value for arginine is 10.76. And the 10.76 kind of gives you an idea of the charges, or gives you an idea of how this structure is made. So down here at the bottom, I'm going to put a general statement. And the general statement is higher the pi value, the closer to the negative charge. Okay, so if I take a look at the structure for arginine, let's draw that structure. COO minus, and then we see an NH3 plus, and then we see a hydrogen, and then we see arginine, which is CH2, 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 NHC double bond NH2 positive and then NH2 again. Okay, so there's the structure for arginine. Ene, think of amine. I see NH2 groups on that molecule as far as the side chain goes. Take a look at those charges that are there. I mean, we got a positive on the NH3 group here, and we've got a positive on the NH2 group here. We've taken a look at the pi value. We can calculate the pi value and know that this pi value is 10.76. Okay, well, how did we do that? Well, you go to that chart that has all the pKa's listed. And then when you look at arginine, you will see that this one has a pKa of 2.17. You'll see that this one has a pKa of 9.04, and then you'll see that this end has a pKa of 12.48. So you should take the two closest in value, 904 and 1248, do an average. You get about 10.76 when you do that. So the general rule of thumb is that if you look at this structure, this is more positive, so therefore it will be attracted to the negative end. So on a sheet of paper, if this is what I've got, what I would see is the negative end would be over here and the positive end would be over here. And if we had to look at the order of separation, I look at the pi values. I would see arginine that would be closest to the negative. So this is arginine. And let's test you. Three-letter abbreviation for arginine is ARG. And one-letter abbreviation for arginine is R. Yeah, the R. So there's arginine, closest to the negative. What would come next would be alanine. Alanine has the next kind of highest pi value, so therefore it's more positive than the others. So alanine would come next, and the one-letter abbreviation for alanine, that's the easy one. That's A, three-letter is ALA. And then what would come next would be our aspartate. And our aspartate has the lowest pi value, 2.98, which means it's more negative than the others, which means it goes toward the positive end chamber more than the others. So this is aspartate, and aspartate is of course ASP, and the one-letter abbreviation is AD. So arginine would be closest to the negative, alanine would kind of be in the middle, and aspartate would be the one closest to the positive charge. So this is why we talk about pi value. It helps us kind of determine the order in which they will separate if I put them in an electrophoresis chamber. And that's what electrophoresis is going to do. Now again, DNA electrophoresis is a little bit different. The basic theory is still there, but the actual diagram and schematic and the way that we explain how these things begin to migrate and separate will be different. But this is electrophoresis at its heart. So look at the pi values. The highest pi value will migrate toward the negative end of the terminal, and then they all kind of follow in order after the fact. So that's how you determine the order of separation when it concerns electrophoresis. Don't be surprised if an essay question on the test will be, explain electrophoresis and how it relates to protein separation or amino acid separation. And this is what you would do. This is what you would describe. You would go through, talk about what electrophoresis is, talk about the different charges in the chamber, talk about spotting the amino acids and how they move and the direction that they do move in. So this is electrophoresis of amino acids. In the next video, we'll take a look at TLC. There's not too much that we have to say there, as many of you have been exposed to TLC before, but for those of you that have not, and may be out of order with the program, then you can at least know overall a general idea of the TLC chamber, what it does, and how the separation exists. So stay tuned. We'll look at part 2 next.